In my bio lab, we made our own independent study, so I chose to do bacteria. I took E. coli in a swab from the shared desktop computers and put them on agar plates and put each of them in 23 degrees Celsius, 25 degrees Celsius, 37 degrees Celsius, and 55 degrees Celsius. I chose different temperatures because I wanted to see which of the bacteria will thrive in the correct temperature. Since the body is at 37 degrees Celsius, it gives me a slight idea on how fast bacteria or germs spread and make us sick. This experiment also gave me insight to how fast the immune system fights back. For today, I will show you my 37 degrees Celsius E. coli and swab. As you can tell, E. coli had the most growth after a day, and the desktop didn't really have much yet. But by day 2, E. coli has spread even more, and you start seeing little white dots on the desktop. On day 3, the E. coli swab lines started to spread out in width. And then on the desktop swab, more bacterial cultures appeared, and some were different colors. On the 4th day, the E. coli became a lot more opaque and did not have sharp edges. The desktop swab, the bacterial cultures got whiter and a few more cultures popped up. On the 5th day, I was not able to tell where my swab lines were. On the 4th day, in person, I could tell that yes, I made this mark right here, but the lines started to run together on the 5th day. As for the desktop swab, all the cultures got whiter and some were changing color, and the one in the center started to look veiny and shriveled up. The E. coli did not grow in length too much, but it did get a little bit wider. And for the desktop agar plate, you can see the bacterial culture in the middle got bigger and browner, and it looks more veiny on the back. And the other bacterial cultures became more vibrant in colors. So how does my experiment relate to class? Well, they're able to relate to chapters 23, 28, and 26. My bacterial growth experiment relates to chapter 23 because every organism, including bacteria, has a taxon. Even though I could not identify my swab bacteria, I had E. coli bacteria growing in my agar dishes. The taxonomic rates go from very broad and big to very small. So the domain is bacteria and the species which is specific is E. coli. What else was mentioned in chapter 23 was phylogenies. Phylogenies is an evolutionary history of what organisms, including which species are closely related and in what order related species evolved, often represented in the form of an evolutionary tree. This evolutionary tree, or phylogeny, is of E. coli, and bacteria in general have a large phylogeny. My experiment also connects to chapter 28. Due to the shape and structure of bacteria, it can show us how resilient it is to different environments. The structure of E. coli bacteria is rod-shaped, and it has a flagella which provides movement. One of the major protectants of the bacteria in a harsh environment such as heat and cold temperatures is called an endospore. For one of my agar plates, I placed it in 55 degrees Celsius and all the bacteria died in it. So that told me that the bacteria on the desktop computer did not have any protective layers that can protect it from heat. The final chapter that relates to my topic of bacteria is chapter 26. It relates to my experiment because it describes the origin of life and which time period did life first appear. It was proven through a microfossil that bacteria was one of the first organisms that appeared on Earth. They assume the fossil dates back to 2.5 billion years ago during the Archean eon. The Archean eon was when life first appeared, And way after the Archaic Eon was the Cambrian explosion, which is when we start to see life thrive on Earth. And during this time, more varieties of bacteria started to show up too.